The journey of food through your body is an incredible process, all thanks to your gastrointestinal or GI tract. This flexible, muscular tube is essentially a pathway that starts at your mouth and ends at the anus, working tirelessly to break down what you eat and absorb vital nutrients. It begins in your mouth, where your teeth and tongue work together to physically break down food, while salivary glands release enzymes to start the chemical digestion of carbohydrates. Once swallowed, the food travels down your esophagus, a muscular tube that uses wave-like contractions called peristalsis to push it towards your stomach. In the stomach, powerful acids and enzymes further break down the food, turning it into a creamy paste. From there, it moves into the small intestine, a long coiled tube where most of the nutrient absorption happens. The liver, pancreas, and gallbladder are accessory organs that play crucial roles here, releasing digestive juices and bile to aid in this process. The small intestine's walls are lined with tiny, finger-like projections called villi, which increase the surface area for efficient nutrient uptake into your bloodstream. What's left then moves into the large intestine, where water is absorbed, and indigestible residue is consolidated into feces. Finally, the rectum stores the feces until they are eliminated from the body through the anus. This entire system works to perform essential functions: ingestion, the selective intake of food; digestion, the mechanical and chemical breakdown of food; absorption, the uptake of nutrients; compaction, absorbing water and forming feces; and defecation, the elimination of waste. It's a complex and vital system, ensuring your body gets the energy and building blocks it needs to thrive. The journey of food through our digestive system is a fascinating and complex process, driven by what we call gastric movement and food propulsion. It all happens in 3 distinct stages: the propulsion stage, the grinding stage, and the retropulsion stage. First, in the propulsion stage, as food enters and fills the stomach, its walls begin to stretch, simultaneously triggering the secretion of gastric juices. The 3 muscle layers of the stomach wall then become active, compressing the food and physically breaking it apart. This is all happening while continuously mixing it with gastric juice, forming a thick fluid called chyme. This chyme mixture is repeatedly folded on itself and mixed until it achieves a uniform texture. Once adequately mixed, a rippling peristalsis begins in the upper half of the stomach, moving the chyme towards the pylorus. Next, we move into the grinding stage. As the food approaches the pyloric valve, the contractions increase in force, effectively grinding the food into chyme. The pylorus can hold about 30 milliliters of chyme and allows only small amounts to pass through the pyloric sphincter. With each contraction of the stomach muscle, approximately 3 milliliters or less of chyme is squirted into the small intestine. Finally, there's the retropulsion stage. Here, the contraction closes the pyloric valve, and the remaining chyme is propelled backward into the stomach for further mixing. This backward and forward movement is crucial for thorough digestion. When the duodenum is filled with chyme and its wall stretches, a nervous reflex called the enterogastric reflex is triggered. This reflex effectively halts gastric activity by inhibiting the vagus nerve and tightening the pyloric sphincter. This entire process ensures that the stomach empties gradually, typically taking about 4 hours to complete. Vomiting is the forceful expulsion of stomach and intestinal contents through the mouth. It's a complex process orchestrated by the emetic center in your brain, specifically in the medulla oblongata. This reflex can be triggered by various factors, including overstretching of the stomach or duodenum, chemical irritants like alcohol or bacterial toxins, visceral trauma, intense pain, or even psychological and sensory stimuli such as repugnant sights, smells, or thoughts. Often, vomiting is preceded by a feeling of nausea and retching. Retching involves coordinated muscular actions. Your thoracic cavity expands and your abdominal muscles contract, creating a pressure difference that dilates the esophagus. The lower esophageal sphincter relaxes, while the stomach and duodenum contract spasmodically. This can be accompanied by a rapid heartbeat, excessive salivation, and sweating. The actual act of vomiting occurs when strong abdominal contractions and rising thoracic pressure force open the upper esophageal sphincter. The esophagus and stomach relax, and the chyme is powerfully ejected from the stomach and mouth, aided by reverse peristalsis in the gastric antrum and duodenum. A distinct and often alarming type is projectile vomiting, characterized by its sudden onset and forceful expulsion without prior nausea or retching. This can be a symptom of neurological issues, but it's also commonly observed in infants after feeding. Our stomachs are incredible organs, designed to break down food with powerful acids and enzymes. But have you ever wondered how it protects itself from this harsh environment? It's a critical question, because without proper protection, our own digestive juices would start to digest the stomach lining itself. The good news is, your stomach has developed 3 ingenious ways to prevent this from happening, ensuring it can do its job without self-destruction. First, let's talk about the mucus coat. The stomach lining is covered in a thick, highly alkaline mucus. This isn't just any mucus. It's a robust barrier that resists the action of both acid and enzymes. Think of it as a protective shield, constantly neutralizing any acid that comes into direct contact with the stomach wall, and preventing corrosive substances from reaching the delicate epithelial cells underneath. Next, we have tight junctions. The epithelial cells that form the stomach lining are not loosely arranged. They are tightly joined together by what we call tight junctions. These junctions act like a strong seal, preventing gastric juice from seeping between the cells and reaching the underlying connective tissue, which would otherwise be vulnerable to digestion. Finally, there's the remarkable process of epithelial cell replacement. The cells lining your stomach have a tough life, constantly exposed to a harsh environment. Because of this, they have a very short lifespan, typically only living for 3 to 6 days. After this short period, they are sloughed off into the chyme and digested right along with your food. But here's the amazing part. These cells are replaced just as rapidly through continuous cell division in the gastric pits. This constant regeneration ensures that any minor damage is quickly repaired, maintaining the integrity of the stomach lining. These 3 mechanisms, the protective mucus coat, the tight junctions between epithelial cells, and the rapid cell replacement, work together seamlessly to create a formidable defense system. They allow your stomach to effectively break down food without breaking itself down in the process. It's a testament to the incredible design of the human body.
Imagine the liver as a landscape of countless tiny cylinders called hepatic lobules. At the center of each lobule is a central vein, with plates of cuboidal hepatocytes radiating outward like spokes on a wheel. Between these plates run blood-filled channels called hepatic sinusoids, where specialized stellate macrophages patrol, removing bacteria and debris. Blood enters each lobule through branches of the hepatic artery and portal vein, both found in the portal triad at the lobule's edge, along with a bile duct. Hepatocytes secrete bile into narrow canaliculi, which channel it toward bile ducts between lobules, eventually forming the right and left hepatic ducts. Your liver produces about 600 to 1,000 milliliters of bile every day. This yellow-green fluid is essential for breaking down and absorbing fats, as well as removing waste products like bilirubin from your blood. Bile secretion happens in 2 main stages. First, hepatocytes in the liver create bile rich in acids, cholesterol, and organic compounds, releasing it into tiny channels called bile canaliculi. Next, bile travels through ducts, either flowing straight into the duodenum or being stored in the gallbladder for later use. Want to master more about liver functions? Keep exploring.
The gallbladder, a small pear-shaped organ nestled beneath your liver, plays a crucial role in your digestive system. Its main job is to store and concentrate bile, a yellowish-green fluid produced by the liver. Think of it as a small reservoir for this powerful digestive juice. Bile is essential for breaking down fats in the food you eat. When you consume fatty foods, hormones signal the gallbladder to contract, releasing concentrated bile into your small intestine. This bile then emulsifies the large fat globules, essentially breaking them into smaller droplets, making it easier for digestive enzymes to do their work, and for your body to absorb those fats and fat-soluble vitamins. The color of bile changes as it's processed. When first secreted by the liver, it's a light yellow-green. However, as it's concentrated in the gallbladder, it becomes a deeper green. Bile also helps your body eliminate waste products, such as bilirubin, which is a byproduct of red blood cell breakdown. Bilirubin is the principal bile pigment and is derived from the decomposition of hemoglobin. Bacteria in the intestine metabolize bilirubin into urobilinogen. Your kidneys then convert some of this urobilinogen into urobilin, which gives urine its characteristic yellow color. The urobilinogen that remains in the intestine is converted to stercobilin, giving feces their brown color. Sometimes, bile can become excessively concentrated, leading to the formation of gallstones. These are hard deposits that can vary in size and are typically made of cholesterol or calcium bilirubinate. While many people with gallstones experience no symptoms, they can cause significant pain and lead to complications if they block the bile ducts. The pancreas is a fascinating organ, primarily known for its exocrine function, which is crucial for digestion. About 99% of the pancreas is made up of exocrine tissue, responsible for producing a significant amount of pancreatic juice daily, around 1,200 to 1,500 milliliters. This pancreatic juice is an alkaline concoction, rich in water, enzymes, zymogens, sodium bicarbonate, and other electrolytes. The acini, which are clusters of cells within the pancreas, are tasked with secreting the digestive enzymes and zymogens. Meanwhile, the ducts within the pancreas handle the secretion of sodium bicarbonate. This bicarbonate is vital because it buffers the hydrochloric acid, HCl, that arrives from the stomach, creating an optimal environment for the pancreatic enzymes to work effectively in the small intestine. Among the key pancreatic zymogens are trypsinogen, chymotrypsinogen, and procarboxypeptidase. These are inactive forms of enzymes that prevent the pancreas from digesting itself. When trypsinogen makes its way into the intestinal lumen, it's converted into its active form, trypsin, by an enzyme called enteropeptidase, also known as enterokinase. Trypsin then plays a crucial role in activating other zymogens, initiating a cascade of protein digestion. Today, we're diving into the fascinating world of how your body regulates pancreatic juice and bile secretion, a crucial process for digestion. There are three main players responsible for this intricate dance. First up is acetylcholine, or ACH. This neurotransmitter comes from your vagus nerves and enteric neurons, and it's a real go-getter. Even before you swallow your food, during what we call the cephalic phase of gastric control, ACH gets the pancreatic acini to start secreting their digestive enzymes. Think of it as your body getting a head start on digestion. Next, we have cholecystokinin, or CCK. This hormone is secreted by the mucosa of your duodenum and proximal jejunum, primarily in response to fats in your small intestine. CCK has a powerful stimulatory effect on your gallbladder, causing it to contract and release bile into the duodenum. It also relaxes the hepatopancreatic sphincter, ensuring that bile can flow freely to help break down those fats. Finally, let's talk about secretin. Your small intestine produces secretin when it detects acidity from the chyme arriving from your stomach. Secretin's main job is to stimulate the ducts of both your liver and pancreas to secrete a rich sodium bicarbonate solution. In the pancreas, this bicarbonate solution flushes the enzymes into the duodenum. This is super important because sodium bicarbonate acts as a buffer, neutralizing the hydrochloric acid coming from your stomach. This neutralization protects your intestinal lining from the harsh acid and creates the optimal pH for digestive enzymes to do their job effectively. The carbonic acid formed then breaks down into carbon dioxide and water, with the CO2 being absorbed into your blood and eventually exhaled. So these three stimuli, acetylcholine, cholecystokinin, and secretin, work together in a beautifully coordinated effort to ensure your digestion runs smoothly. The small intestine is a marvel of biological engineering, perfectly designed for its crucial role in nutrient digestion and absorption. Its inner lining, or mucosa, is composed of simple columnar epithelium and boasts an incredibly large internal surface area, essential for efficient processing of the food we eat. This vast surface area is achieved through three distinct types of folds and projections: circular folds, villi, and microvilli. First, let's talk about circular folds. These are transverse to spiral ridges, up to a centimeter high, found in the mucosa and submucosa. They're not just there for show. These folds slow down the chyme, the partially digested food, and guide it along a spiral path. This increased contact with the mucosal surface promotes more thorough mixing and nutrient absorption. Circular folds begin in the duodenum, become particularly large and closely spaced in the jejunum, and then gradually diminish in size and density in the ileum. This progression directly correlates with the amount of nutrient absorption that occurs in each region. Next, we have the villi. These finger-like projections give the inner lining of the intestine a fuzzy, terry cloth-like texture. Each villus is about 0.5 to 1 millimeter high and is largest in the duodenum, becoming progressively smaller further down the intestine. Villi are covered with two types of epithelial cells: columnar enterocytes, which are the primary absorptive cells, and mucus-secreting goblet cells. The core of each villus contains a rich network of blood capillaries, which absorb most nutrients. and a lymphatic capillary called a lacteal, specifically designed for absorbing most lipids. Finally, we come to the microvilli. These are much smaller, plasma membrane extensions, about 1 micrometer high, that form a fuzzy brush border on the surface of each enterocyte. Beyond further increasing the surface area for absorption, microvilli are home to crucial brush border enzymes. These enzymes are responsible for the final stages of chemical digestion. Unlike enzymes secreted into the lumen, these require direct contact with the chyme for digestion to occur, a process aptly named contact digestion. Together, these three structures, circular folds, villi, and microvilli, work in concert to create an astonishingly vast surface area, maximizing the small intestine's ability to extract every possible nutrient from our food. Hey everyone, let's talk about something fundamental to our digestion: intestinal motility. It's how your small intestine gets things done, performing three crucial functions. First, it mixes chyme. That's the partially digested food from your stomach with essential intestinal juice, bile, and pancreatic juice to break it down further. Second, it churns that chyme, bringing it into close contact with the intestinal lining for what we call contact digestion. And most importantly, nutrient absorption. And third, it moves any leftover residue towards your large intestine for eventual elimination. Now, how does it do all this? One key movement is called segmentation. Imagine stationary, ring-like constrictions appearing and relaxing at various points along your intestine, constantly kneading and churning the contents. This is the most common type of intestinal contraction, driven by pacemaker cells in your muscularis externa. These cells set a rhythm, about 12 contractions per minute in the duodenum and 8 to 9 in the ileum. While segmentation primarily mixes, it also causes a slow, steady progression of chyme towards the colon. But for more directed movement, we have peristalsis. These are the classic wave-like contractions that actively propel food forward. Circular muscles behind the food contract, pushing it along, while longitudinal muscles ahead relax, making way for smooth passage. The duodenum even secretes a hormone called motilin, which triggers a peristaltic wave. This wave travels a certain distance, dies out, and then another wave begins a little further down the tract. These successive, overlapping waves are known as the migrating motor complex. They effectively milk the chyme towards the colon over about a 2-hour period. A second complex then sweeps out any remaining residue and bacteria, helping to prevent bacterial overgrowth. Interestingly, when you eat your next meal and your stomach refills, peristalsis is suppressed, and segmentation kicks back in as new chyme enters the small intestine. It's a beautifully coordinated system, constantly working to digest your food and absorb those vital nutrients. Today, we're diving into the fascinating world of carbohydrate digestion and absorption, a vital process that fuels our bodies. It all begins with the most digestible dietary carbohydrate, starch. Starch is first broken down into oligosaccharides, which can be up to 8 glucose residues long. These are then further digested into maltose, and finally into individual glucose molecules, which are readily absorbed by the small intestine. The initial breakdown of starch actually starts in your mouth, where salivary amylase begins to chop it into shorter segments. Interestingly, about half of the starch you consume is digested before it even reaches your small intestine. The digestion process then picks up again in the small intestine, when the chyme, the partially digested food from your stomach, mixes with pancreatic amylase. This powerful enzyme ensures that starch is entirely converted into oligosaccharides and maltose within a mere 10 minutes. To complete the digestion of these smaller carbohydrate units, two crucial brush border enzymes, dextranase and glucoamylase, hydrolyze the oligosaccharides. Meanwhile, maltase specifically breaks down maltose. Beyond starch, our diets also contain major disaccharides like sucrose, commonly known as cane sugar, and lactose, or milk sugar. These are handled by other brush border enzymes. Sucrase digests sucrose into glucose and fructose, and lactase breaks down lactose into glucose and galactose. These resulting monosaccharides, glucose, fructose, and galactose, are the final products ready for absorption. Now, let's talk about how these vital monosaccharides get into your bloodstream. The plasma membrane of the enterocytes, the cells lining your small intestine, are equipped with specialized transport proteins for this very purpose. A significant portion, about 80% of the absorbed sugar is glucose, which is actively taken up by a sodium glucose transporter, or SGLT. When sugar enters the extracellular fluid, it increases its osmolarity, which in turn draws water from the intestinal lumen through the now leaky tight junctions between the epithelial cells. This water carries even more glucose and other nutrients with it through a process called solvent drag. After a high-carbohydrate meal, solvent drag can actually absorb 2 to 3 times as much glucose as the SGLT transporters alone. The SGLT also absorbs galactose, while fructose is absorbed by facilitated diffusion using a separate carrier protein. Once inside the enterocyte, most of the fructose is converted into glucose. Finally, glucose and galactose are transported out of the base of the cell by facilitated diffusion, and are then absorbed by the blood capillaries of the villus. From there, the hepatic portal system delivers these absorbed sugars directly to your liver, ready to be used for energy or stored for later. Let's talk about protein digestion and absorption, a fascinating journey our food takes. The amino acids our small intestine absorbs actually come from 3 main places. First, there are the dietary proteins we eat every day. Then, surprisingly, our own digestive enzymes get digested by each other. And finally, sloughed epithelial cells, which are digested by these very same enzymes, also contribute. Now, the enzymes responsible for breaking down proteins are called proteases, or sometimes peptidases. Interestingly, no protein digestion actually happens in your mouth, because you won't find any proteases in your saliva. The real work begins in the stomach. Here, an enzyme called pepsin gets to work, hydrolyzing specific peptide bonds and breaking down proteins into smaller polypeptides. But the small intestine is where the magic truly happens. Pancreatic enzymes like trypsin and chymotrypsin take over, further hydrolyzing those polypeptides into even shorter oligopeptides. From there, 3 more specialized enzymes meticulously break them down 1 amino acid at a time. Carboxypeptidase removes amino acids from one end of the chain, while aminopeptidase tackles the other end. And for those last 2 amino acids, dipeptidase steps in to split dipeptides right in the middle, releasing them as free amino acids. Before their amino acids are released into the bloodstream, dipeptides and tripeptides are hydrolyzed within the enterocytes. Once they're ready, they leave the cell through facilitated diffusion, enter the capillaries of the villus, and are then carried away in the hepatic portal circulation, ready to be used by your body. It's a complex and efficient process that ensures we get all the building blocks we need. Today, we're diving into the fascinating world of lipid digestion and absorption. Essentially, how your body breaks down and uses the fats you eat. The process starts in your mouth, where lingual lipase from your salivary glands begins the initial breakdown. Next, the food moves to the stomach. Here, gastric lipase takes over, digesting about 10% to 15% of dietary fat. The stomach also emulsifies the fat, breaking it into smaller droplets to dramatically increase the surface area for enzyme action. These emulsified droplets then head to the small intestine, where they are coated by bile components like lecithin and bile acids. The real heavy lifting occurs here, thanks to pancreatic lipase. This powerful enzyme breaks fats down into 2 free fatty acids and 1 monoglyceride, capable of digesting your daily fat intake in just minutes. Absorption relies on tiny bile droplets called micelles. Think of micelles as microscopic taxis. They pick up fat-soluble vitamins, cholesterol, and the digestion products. Because micelles are hydrophilic, they move freely through the watery small intestine to reach the enterocytes, the cells lining the intestine. Upon arrival, they release their lipid cargo, which diffuses into the cells, while the micelles are reused. Inside the enterocytes, the fatty acids and monoglycerides are reassembled into triglycerides within the smooth endoplasmic reticulum. The Golgi complex then steps in, combining these triglycerides with cholesterol and coating the package with phospholipids and proteins. This new structure is called a chylomicron. Chylomicrons are packaged into secretory vesicles and released from the cell. Since they are too large to enter the bloodstream directly, they take a detour through porous lacteals in the lymphatic system. From there, they travel through the lymph and eventually enter your bloodstream, ready to be used or stored. It's a complex, efficient system, ensuring your body gets the vital fats it needs. Next, we're diving deep into the fascinating world of the large intestine. Let's start with its microscopic characteristics. The large intestine's lining, or mucosa, features a simple columnar epithelium throughout most of its length. However, as we approach the lower half of the anal canal, this changes to a non-keratinized, stratified squamous epithelium. This specialized tissue is crucial for resisting the abrasion caused by the passage of feces. You won't find circular folds or villi here, unlike in the small intestine. Instead, the large intestine is home to intestinal crypts. These crypts are deeper than those found in the small intestine and boast a higher density of goblet cells. These goblet cells are vital because mucus is their primary and most significant secretion, playing a key role in lubrication and protection. Both the lamina propria and submucosa of the large intestine are rich in lymphatic tissue. This abundance of lymphatic tissue provides essential protection against the vast number of bacteria that densely populate this part of our digestive system. Moving on to the incredible world of intestinal microbes and gas. Our large intestine is a bustling ecosystem, harboring approximately 800 species of bacteria, collectively known as the gut microbiome. We share a mutually beneficial relationship with many of these microorganisms. They perform crucial functions for us, such as digesting cellulose, pectin, and other plant polysaccharides, compounds for which our bodies lack the necessary digestive enzymes. We then absorb the resulting sugars. Some of these beneficial bacteria also synthesize essential B vitamins and vitamin K, which are subsequently absorbed by the colon. Now, let's talk about intestinal gas. While much of it is swallowed air, our gut microbes also contribute significantly to its production. The large intestine typically contains about 7 to 10 liters of gas, with approximately 500 milliliters expelled daily as flatus. This flatus is primarily composed of nitrogen, carbon dioxide, hydrogen, and methane. It also contains hydrogen sulfide and two amines, indole and scadole. It's these compounds, indole, scadole, and hydrogen sulfide, that are largely responsible for the characteristic odor of flatus and feces. It's a complex and vital part of our digestive health. Let's talk about the incredible journey of food through your large intestine, a process that takes a surprising 36 to 48 hours to transform meal residue into what we call feces. The longest part of this trip, about 24 hours, happens right in your transverse colon. During this time, your colon is a master of reabsorption, pulling out vital water and electrolytes like sodium chloride. What's left, the feces, is typically about 75% water and 25% solids. And those solids? They're a fascinating mix. Around 30% bacteria, another 30% undigested dietary fiber, 10 to 20% fat, and then smaller amounts of protein, shed epithelial cells, salts, mucus, and other digestive secretions. But how does all this move along? Enter haustral contractions. These are a common type of colonic motility, a kind of segmentation that happens roughly every 30 minutes. Think of it as a gentle churning and mixing, which is crucial for promoting water and salt absorption. Then, things get a bit more intense with mass movements. These are much stronger contractions, occurring just 1 to 3 times a day, each lasting about 15 minutes. They're designed to move residue several centimeters at a time, and are often triggered by reflexes, like the gastrocolic and duodenocolic. You'll typically experience these mass movements in the transverse to sigmoid colon, often within an hour after breakfast, as your body clears out the feces that have accumulated overnight and stretched the colon. Finally, we reach defecation. This process is initiated when the rectum stretches, stimulating the defecation reflexes. There are 2 main reflexes at play. First, the intrinsic defecation reflex, which is entirely mediated by the myenteric plexus. Stretch signals travel through this plexus to the muscularis of the descending and sigmoid colon and the rectum, activating a peristaltic wave that pushes feces downward and relaxes the internal anal sphincter. This reflex, however, is relatively weak. The more powerful player is the parasympathetic defecation reflex, a spinal reflex. Here, stretch signals are sent to the spinal cord, and motor signals return via the pelvic nerves. These signals intensify peristalsis in the descending and sigmoid colon and rectum, while also relaxing the internal anal sphincter, making the final expulsion possible. Welcome to the incredible journey of food through your body. It all begins in your mouth, where the mechanical breakdown of food kicks off. Your teeth physically break down food into smaller particles through chewing. a process called mastication. As you chew, your food mixes with saliva. This isn't just for moistening. Salivary amylase, an enzyme in your saliva, starts the chemical digestion of starch, breaking it down into maltose. While saliva is always present to keep your mouth moist, a much larger amount is released from your salivary glands when food enters. Interestingly, no food absorption happens here. After the mouth, food moves into the pharynx and esophagus, which act as simple passageways, guiding food to its next destination, the stomach. The pharynx, a muscular funnel, connects your oral cavity to the esophagus and your nasal cavity to the larynx. It's a crucial intersection for both your digestive and respiratory tracts. Its muscular layers, particularly the superior, middle, and inferior pharyngeal constrictors, work to force food downward during swallowing. When you're not swallowing, the inferior constrictor stays contracted, preventing air from entering your esophagus. This acts as the upper esophageal sphincter. Next up is the esophagus, a straight muscular tube about 25 to 30 centimeters long. It starts between vertebra C6 and the cricoid cartilage behind your trachea, at the upper esophageal sphincter. The submucosa of the esophagus contains glands that secrete lubricating mucus, making the journey smoother for your food. It passes through your mediastinum, then penetrates the diaphragm at the esophageal hiatus, continuing a few more centimeters to meet your stomach at the cardial orifice. Here, food briefly pauses at the lower esophageal sphincter, or LES. This vital sphincter prevents stomach contents from regurgitating back into the esophagus, protecting its lining from erosive stomach acid, which is what causes that burning sensation we call heartburn. Today, we're diving into the fascinating world of how we eat, specifically focusing on food ingestion and the incredible process of swallowing. It's something we do every day without a second thought, but it's actually a complex dance involving over 22 muscles in your mouth, pharynx, and esophagus. All of this is precisely coordinated by a special control center in your brainstem called the medulla oblongata. Swallowing, or deglutition, happens in 3 distinct phases. First up is the oral phase, and this one is entirely under your voluntary control. As you chew, your tongue is hard at work, collecting the food, pressing it against the roof of your mouth to form what we call a bolus. That's a soft mass of chewed food. Then it pushes this bolus towards the back of your mouth. The food gathers in your oral pharynx, right in front of the epiglottis. Once that bolus reaches a certain size, the epiglottis tips back, and the food slides around it, through a space on each side and into your laryngopharynx. Next, we move into the pharyngeal phase, and this is where things become involuntary. Your body takes over. The soft palate and the root of your tongue work together to block food and drink from going up into your nasal cavity or back into your mouth. To prevent choking, your breathing automatically pauses. The epiglottis covers your laryngeal opening, and your vocal cords close off your airway. These actions also widen the upper part of your esophagus, getting it ready to receive the food. Then, the pharyngeal constrictor muscles contract in a precise sequence, pushing the bolus downward into your esophagus. Finally, we reach the esophageal phase, characterized by a wave of involuntary contractions called peristalsis. This is controlled by both your brainstem's swallowing center and a network of nerves in your esophageal wall. The circular muscle layer above the bolus tightens, pushing the food down. Below the bolus, the circular muscle relaxes, while the longitudinal muscle contracts, pulling the esophageal wall upward and dilating it to receive the descending food. While gravity helps when you're upright, peristalsis is the real hero, ensuring you can swallow no matter your body's position, even standing on your head. Liquids typically reach your stomach in just 1 to 2 seconds, and a food bolus in 4 to 8 seconds. As the bolus nears the end of the esophagus, the lower esophageal sphincter relaxes, allowing it to pass into your stomach. It's a truly amazing process, isn't it? Welcome to the stomach's digestive glands. Our stomach is lined with a special kind of tissue called a simple columnar glandular epithelium. This lining isn't smooth. It forms prominent folds known as gastric rugae. Delving deeper, the lamina propria, a layer beneath the epithelium, is almost entirely filled with tubular glands. These aren't just random indentations. The gastric mucosa is dotted with depressions called gastric pits, all lined with that same columnar epithelium. Imagine tiny openings, and at the bottom of each pit, 2 or 3 tubular glands extend downwards, spanning the rest of the lamina propria. Now, these glands aren't uniform throughout the stomach. In the regions closest to the esophagus and the small intestine, the cardial and pyloric regions, they're called cardial glands and pyloric glands respectively. In the main body of the stomach, they're known as gastric glands. Let's break down the cellular composition of these digestive glands, as each cell type plays a crucial role. First, we have mucus cells. These are the primary secretors of mucus, especially abundant in the cardial and pyloric glands. In the gastric glands, they're specifically called mucus neck cells and are concentrated in the narrow neck of the gland, where it opens into the gastric pit. This mucus is vital. It forms a protective layer, shielding the stomach lining from its own potent acids and enzymes. Next, we encounter regenerative stem cells. These essential cells are found at the base of the pits and the neck of the glands. They divide rapidly, constantly producing new cells to replace those that wear out. These newly generated cells migrate both upwards to the gastric surface and downwards into the glands, ensuring a continuous supply of fresh cells. Parietal cells are another key player, found mostly in the upper half of the gland. These remarkable cells secrete hydrochloric acid, HCl, which is crucial for breaking down food and activating digestive enzymes. They also produce intrinsic factor, which is essential for vitamin B12 absorption, and an appetite-stimulating hormone called ghrelin. While primarily found in gastric glands, a few can also be found in the pyloric glands. Then there are chief cells. These cells dominate the lower half of the gastric glands and are responsible for secreting the enzymes gastric lipase and pepsinogen. Pepsinogen, once activated by hydrochloric acid, becomes pepsin, which breaks down proteins. Chief cells are absent from the cardial and pyloric glands. They are named chief because they are the most numerous cell type in these glands. Finally, we have enteroendocrine cells. These specialized cells secrete hormones and paracrine messengers that regulate digestion. They are most abundant in the gastric and pyloric glands. Let's talk about gastric secretions, the amazing fluids our stomach produces every day. Our gastric glands churn out about 2 to 3 liters of gastric juice daily, and it's mainly composed of water, hydrochloric acid, and pepsin. Hydrochloric acid, or HCl, is incredibly potent, giving gastric juice a pH as low as 0.8. Specialized parietal cells use an active transport protein called H+-K+ ATPase to pump hydrogen ions into the stomach's lumen. Meanwhile, bicarbonate ions are exchanged for chloride ions from our blood plasma. This clever system means HCl builds up in your stomach while bicarbonate ions accumulate in your blood. So, what does this powerful acid do? It activates enzymes like pepsin and lingual lipase, breaks down connective tissues and plant cell walls to help liquefy food into chyme, and even converts ingested ferric ions to ferrous ions. Plus, it's a crucial part of our innate immunity, destroying most pathogens we ingest. Next up is pepsin. Many digestive enzymes start as inactive proteins called zymogens. In the stomach, chief cells secrete pepsinogen, a zymogen that HCl then converts into active pepsin by removing some amino acids. Pepsin's job is to digest dietary proteins into shorter peptide chains, which then move to the small intestine for further digestion. Don't forget gastric lipase. Chief cells also secrete this enzyme, which, along with lingual lipase, plays a minor role in digesting about 10% to 15% of the dietary fat in your stomach. And then there's intrinsic factor. Parietal cells secrete this vital substance, essential for your small intestine to absorb vitamin B12. This secretion only happens in the stomach, which is why if someone has their stomach removed, they need vitamin B12 injections or oral supplements. As we age, intrinsic factor secretion can decrease, increasing the risk of anemia, especially pernicious anemia. Finally, let's touch on chemical messengers. The gastric and pyloric glands contain various enteroendocrine cells that produce up to 20 hormones and paracrine signals. Many of these are peptides found in both the digestive tract and the central nervous system, earning them the name gut-brain peptides. These include important players like substance P, vasoactive intestinal peptide, VIP, secretin, gastric inhibitory peptide, GIP, cholecystokinin, CCK, and neuropeptide Y, NPY. It's a complex and fascinating system, all working together to keep our digestion running smoothly. Next, we're diving into the fascinating world of gastric function. a highly regulated process that ensures our bodies efficiently digest food. It's broken down into three main stages: the cephalic, gastric, and intestinal phases. And interestingly, they can all overlap. First up, the cephalic phase. This is where your stomach gets ready for action even before food arrives. Just the sight, smell, taste, or even the thought of food can kickstart this process. Your brain, specifically the hypothalamus and medulla oblongata, receives these sensory inputs and sends signals down the vagus nerve to your stomach's enteric nervous system. This stimulates your parietal cells to release acid and your G cells to produce gastrin, essentially priming your stomach. About 40% of your stomach's acid secretion happens right here, getting everything prepared to process that delicious meal. Next, we move into the gastric phase, which begins once food actually enters your stomach. This is a crucial period where swallowed food and partially digested proteins really get things going. About half of your total acid secretion and 2/3 of all gastric secretion occur during this phase. How does it work? Well, two main ways: the stretching of your stomach as it fills, and a rise in the pH of its contents. Three key chemicals, acetylcholine, histamine, and gastrin, are the primary stimulators here, all working to get those parietal cells secreting hydrochloric acid and intrinsic factor. Your chief cells also get in on the action, releasing pepsinogen. As digestion progresses and peptides leave the stomach, the pH drops. Once it falls below a pH of 2, a negative feedback loop kicks in, inhibiting the parietal and G cells, signaling that the need for pepsin and HCl is decreasing. Finally, we have the intestinal phase, which starts when chyme, that semi-digested food, begins to arrive in your duodenum. This phase is a delicate balance of hormonal and nervous reflexes. The acid and fats in the chyme trigger the enterogastric reflex. Your duodenum sends signals to the medulla, which in turn reduces vagal stimulation of the stomach and stimulates sympathetic neurons to send inhibitory signals. This essentially slows things down. Additionally, enteroendocrine cells in the duodenum release secretin and cholecystokinin, which primarily stimulate the pancreas and gallbladder, but also help to suppress gastric secretion and motility. These cells also secrete glucose-dependent insulinotropic peptide, or GIP, which stimulates insulin secretion. It's a complex and beautifully coordinated system, ensuring your body gets the most out of every meal.